Good morning, everyone. Very nice to be here. I really appreciate the invitation from the organizers. My name is Chunping from Dairy Research Institute, State Key Laboratory of Dairy Biotechnology, Bright Dairy and Food Company. My topic today is biofilm formation risk assessment for psychrotrophic Pseudomonas in raw milk by MALDI-TOF mass spectrometry. Now, please allow me to introduce myself. I'm a professor-level senior engineer in food biotechnology. I graduated from Fudan University with my bachelor and PhD degree in chemistry. Then in 2009, I came to Bright Dairy and Food Company to work as a researcher. And now I am a director of Scientific Research Center and the group leader in dairy safety and probiotics. As for the Institute, State Key Laboratory of Dairy Biotechnology, SKLDB, it's the only one national key laboratory in the Chinese dairy industry. In 2013, it was formally accredited by the Ministry of Science and Technology of China. There are four research aspects. The first one is quality and safety control. The second one is new dairy starters and probiotics. The third one is functional dairy products. And the fourth one is cheese and its byproducts. Now let's move to the background of the presentation. We know that the psychrophile is a bacterium with optimal growth temperature from minus 15 degrees Celsius to 20 degrees Celsius. However, in the dairy industry, it means the bacteria that grow and reproduce at temperatures are not higher than 10 degrees Celsius. So the accurate name of these bacteria should be called psychrotrophs. From November 2012 to December 2013, during the 12 months, we collected the milk samples from 10 dairy farms in Shanghai and around to screen the psychrotrophs. From the distribution of the psychrotrophs, we can see that the top one genus is Pseudomonas. And in the Pseudomonas genus, the top one species is Pseudomonas fluorescens. And then in another 12 months from July 2015 to June 2016, we also collected the milk samples from the 10 dairy farms in Shanghai and around and tested their sequences. We can see that the Pseudomonas have a relatively high abundance in winter. And the psychrotrophs have a relatively high lipase activity and a relatively high protease activity. So it might bring the problems to the milk products like swallow, sour, bitter, and so on, the unpleasant flavor and taste to the products. And it also can affect the colors and the textures and affect the shelf life of ultra-high temperature treated milk directly. There are three risk factors of the psychrotrophs. The first is the bacteria themselves. The second is the heat-resistant enzymes they produce. The third is the biofilms. We know that the biofilms result in the difficult removal of psychrotrophs and promote the secretion of heat-resistant enzyme in Pseudomonas, and also provide growth substrate of pathogenic bacteria and enhance bacterial drug resistance. So it is very important to study the differences in biofilm formation by so many different psychrotrophs, and to evaluate biofilm forming risks of different psychrotrophs. The usually used two methods are genotyping and metabolic phenotyping. We listed the commonly used typing methods. The first 5 are genotyping, and the last one is the MALDI-TOF based on protein analysis. We know that the MALDI-TOF mass spectrometry is easy to operate, with low cost in consumables, high throughput, and high accuracy. So it has been extensively applied in the identification of bacteria species in medical and food research areas, especially, for example, the identification of drug resistance ability of bacteria by profiling bacterial metabolites. So it is promising to use MALDI-TOF MS to assess the biofilm formation ability of psychrotrophic Pseudomonas. We believe that the mass fingerprints of bacterial cells can cover a wide range of biomolecules that can closely associated with the bacterial phenotype. So the main objective of this study is to establish a rapid and reliable method for typing psychrotrophs. Based on the MALDI-TOF technology, the phenotype differences of psychrotrophs were distinguished and their correlation with biofilm forming risk were analyzed. Then we established and validated a classification model of psychrotrophs with different biofilm formation abilities based on the mass spectrometry, fingerprinting, and machine learning methods. Firstly, from the classification of Pseudomonas fluorescens by the phenogenic tree based on aDNA sequence. Although aDNA protein is a transcriptional activator related to biofilm formation in Pseudomonas fluorescens, it is not strongly correlated with biofilm forming ability. Because differences in gene sequences do not mean differences in gene expression. And it is also possible that the strains with later nucleotide differences may have greater phenotypic differences. Secondly, from the relationship between clustering results based on 16S rRNAs sequencing and biofilm forming ability, and the relationship between clustering results based on MLSD data and biofilm forming ability, we can see that the correlation between the typing results and the bioforming ability were not significant. There was no strong relationship between the phenotype of biofilm forming and phylogeny within the species, but the overall trend was consistent. Then how about the mass fingerprinting based on MALDI-TOF? This slide showed the schematic illustration of establishing a classification model for rapid identification of psychrotrophic strains. From this procedure, we can see that it is very simple using the MALDI-TOF MS. From the typing of 31 strains, we can see that the same species of strains were clustered together. The pink ones were the Pseudomonas fluorescens. Therefore, the MALDI-TOF typing could distinguish strains at a species level, firstly. Then we can test the other 6 strains from Tibet sample. Tibet is very west of China, but our method using the samples from Shanghai area, it is very east of China. From the slide, we can see that the 6 strains sampled from Tibet were all accurately identified to species. And then we can see the biofilm forming risks if Pseudomonas fluorescens on this slide. 23 Pseudomonas fluorescens were isolated from the raw milk based on 16S RNA sequencing. We use the 23 Pseudomonas, 15 of them as training sets, and eight of them as test sets. According to the measured OD values, the biofilm forming risks of the isolates were divided into 3 grades. 7 of them are weak, 11 of them are medium, and 5 of them are strong. This slide showed the typical 4 isolates. From the mass spectra, we can see that the 4 isolates have a very high similar mass spectra. Distinctive peaks can be observed among the 4 classes that can differentiate the strong and medium from the weak groups. The peak of 6049 increases in weak groups, while the peaks of others increase in strong and medium groups. The group strong and group medium have similar regulation trends. Through the comparison of the cluster analysis based on ML-STIP typing and MALDI-TOF typing, we can see that the biofilm forming ability of psychrotrophic isolates cannot be well classified by ML-STIP typing, but can be well classified by the MALDI-TOF. The PLSDA plot demonstrated that the training sample can be well discriminated into 2 groups, the strong and medium group and the weak group. The higher VIP scores in PLSDA plot represent more significance, the mass spectrometric features in the classification of the biofilm forming ability. The volcano plot shows that the mass spectrometric features from the training samples of different biofilm forming ability can have varied intensity. For features, the 6030, 6049, the 5125, and the 5682 were more intensive for the isolates in the weak groups, but the other features were more intensive in the strong and medium groups. Then a classification model was built using random forest analysis for the training set and validated on this test set. We can see that the 6 strains were were identified as a strong and medium group, and the other 2 strains were identified as weak groups. It shows 100% accuracy in biofilm forming risk identification. Combining MALDI-TOF molecular mass and fingerprinting and proteomic analysis, we can see that the 6 of the 15 peaks were annotated as proteins. The protein associated with biofilm formation were identified, suggesting the potential of monitoring biofilm forming ability by detecting and tracking the protein markers. Further studies would be performed to assess the reliability of these proteins as biomarkers for biofilm forming. And this is a published paper on LWT in 2023. Finally, I would like to thank our group and our cooperation group, Professor Qiao Liang at Fudan University. And we also would like to thank the funding. Finally, thanks for all of you. Thanks for the attention. Thank you very much.